Hello everybody, in this video lecture we will cover chapter 9 part 2 and this is the central dogma of molecular biology. Okay, so what is the central dogma? Central dogma, the name itself tells you must be something very important in biology. Well, central dogma tells us that inside the DNA we have information, and this is information about protein synthesis. So for your cells, for your liver cells, for your kidney cells, for your neurons, for your skin cells, for your muscle cells, and also for plant cells, for other animal cell, any cellular organism, right? And we have only cellular organisms on our planet. So for any single cell to perform its cellular function, it must have information stored inside the DNA. But this information first needs to be encoded into RNA. So information flows from DNA to RNA, and then your cells using this RNA to make proteins. And proteins, those are molecules that allow your cells to do their cellular work. We have this saying in biology: DNA has all the glory and proteins do all the work. So actually everything your cells do, all those functions depend on different proteins that your cells synthesize. And to synthesize those proteins, they need information stored in the DNA. Right, so that's what this slide tells us. In both prokaryotes and eukaryotes, the second function of DNA, the first was replication. Right, so DNA can replicate, make an identical copy of itself, and this copy of DNA molecule can be passed from cells to cells, from generation to generation. But the second function is to provide information needed to construct proteins. And proteins are necessary so cell can perform all of its function. So you can see that's a molecule of DNA, and DNA in eukaryotic cells stored inside the nucleus. Then through the process transcription, your cell's gonna make RNA, and RNA code for sequence of amino acid that make primary structure. Those sequence of amino acid makes primary structure of the protein. Right? And then proteins folds, assume its final shape, and proteins do their work inside our cells. So that's the central dogma. From DNA to RNA, from DNA to RNA and to proteins. So that's what central dogma tells us. I'm gonna remove all this stuff. Okay, so we have this flow of information from DNA to RNA to protein. So through the processes of transcription and translation, protein is built with a specific sequence of amino acid that was originally encoded in the DNA. This model discusses the details of the transcription, and the central dogma states that DNA encodes RNA, which in turn encodes proteins. And the processes you see here, transcription and translation. So transcription is the process of making RNA molecule from complementary DNA sequence, and translation is making proteins from RNA. So protein synthesis has three phases: transcription, RNA processing, and translation. So here we have our central dogma from DNA to RNA to proteins. And the first is transcription. So here's our eukaryotic cell, And eukaryotic cell has a nucleus, so that's a nucleus. And you can see transcription happens inside the nucleus. So we start with DNA molecule, right? Here's a DNA molecule, and we end up with RNA molecule, but specific RNA molecule that is called messenger RNA. And there is RNA processing that also happen inside the nucleus, because first your cell gonna synthesize pre-messenger RNA, and then it will be modified to make functional RNA. Then RNA will move out of the nucleus to the cytoplasm. This is this molecule over here, right? This is our messenger RNA. And it will attach to a ribosome, and ribosome will synthesize protein using messenger RNA as instruction. So we're talking about transcription. So remember that transcription is we're going from DNA to messenger RNA. So transcription has initiation. Initiation begins when DNA unwound, forming transcription bubble. Enzymes and other protein involved in the transcription bind to the promoter. So here's the double helix of the DNA, and here we have this transcription bubble where we open up the DNA and we use special enzyme, RNA polymerase, the one that synthesizes this RNA molecule. So this is initiation, just when we open up DNA molecule, right, and we start the process. The second stage of transcription, so we're still talking about transcription. The first was initiation, then elongation. We have an enzyme, it's called RNA polymerase, and this enzyme, it moves along the DNA, so that's blue molecule here is a DNA, and uses this DNA as a template to build complementary RNA molecule. So you can see how RNA correspond to DNA. So if we have C over here, then we have G. If we have A on DNA, then we have U on RNA. Remember, RNA has U, not T. So no T. So that's elongation. And then we get to termination. That's the last process of transcription. And transcription stops when RNA polymerase reaches terminator. So it's a special region on a DNA that pretty much sends signal to RNA polymerase to stop transcription. So once gene is transcribed, RNA polymerase is instructed to dissociate from the DNA template and liberate the newly made messenger RNA. Okay, so that was the first step, transcription. The second step is RNA processing. So again, we're back to the same picture. What we already described is transcription. So we are making protein, right? So that's our goal, to make protein. And we're starting with molecule of DNA. So first we transcribed DNA into pre-messenger RNA. And as we already said, transcription has initiation phase, elongation phase, and termination phase. So now we have this pre-messenger RNA, and we're ready to move to the second step, RNA processing. So eukaryotic RNA processing. Eukaryotic genes are composed of protein coding sequences called exons. And some sequences intervening sequences called introns. Introns, they do not encode for functional proteins. That's why they must be sliced out. So the process of removing introns and reconnecting exons is called splicing. So here our RNA processing. You guys can see pre-messenger RNA, and it has exons and introns. And what we need to do, we need to splice out those introns. We need to cut them out, and we have a special enzyme to do it. And then your cells need to connect exons together, making our mature RNA molecule. And that's not it, because what else your cells need to do? They need to add 5' cap and 3' poly A tail. And those are special, molecules that are added to the beginning of the messenger RNA and to the end. Right? And this poly A tail is needed to prevent degradation by enzymes, because your enzymes will chop off part of the messenger RNA, and we don't want them to damage exons, because exons are important in the protein synthesis. Poly A tail, we can afford losing some of these, molecules here. So they're not that important. They are attached to prevent degradation by enzymes. Okay, so here's a question for us. What would be the complementary RNA strand for the following DNA sequence? So we have DNA, right? That is GC, GTA, TG. So it would be like, okay, so here's the DNA molecule, and then your cells unwind it. So it's open it like this, and the DNA is polymer of nucleotides. So here's our nucleotides, and we have 4 different nucleotides. In this example, we have G, C, G, T, A, T, and G, right? G, C, G, T, A, T, G. Right, so RNA polymerase gonna make messenger RNA that will be complementary, right? So we also will have 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7 nucleotides. And because they're complementary, where we have G, we will have C, G, C, A. We cannot have T, that's why it's going to be U, A, and the last one going to be C. Right, so that's going to be messenger RNA. And the last step is translation or making proteins. So we already described transcription and RNA processing that happen inside the nucleus. Now we made this messenger RNA. It's single-stranded. And I don't want it to look like a helix, double helix or something. So messenger RNA leaving the nucleus and attaches itself to ribosome, right over here, and serve as a template for protein synthesis. So now we will talk about translation, and translation happens inside cytoplasm. So translation or protein synthesis machinery will include the large and small subunits of the ribosome, messenger RNA, and tRNA. So here's the large subunit of the ribosome, small subunit of ribosome. This is our messenger RNA that we just synthesized through the process of transcription and RNA processing. And This one is called tRNA or transfer RNA. So ribosome is a complex molecule made of ribosomal RNA molecules and proteins that form a factory for protein synthesis in the cell. So on this diagram, you can see structure of ribosome, large and small subunits. And they have the sites. We have P site, we have A site, we have E site over here. That will be important in our discussion in a second. tRNA or transfer RNAs are molecules that act as temporary carriers of amino acid, bringing the appropriate amino acid to the ribosome. So in this picture over here, you can see a tRNA, and on one side over here we have amino acid attached, and each tRNA has its specific amino acid attached to it. Like this one is serine, another one is tyrosine. And so here's our amino acid attachment site where amino acids are attached, and template recognition site, it's this one. And template recognition site is called anticodon, and it contains a sequence of 3 bases that are complementary to the codon of messenger RNA. So here's a messenger RNA that was synthesized, and it has these codons, and tRNA has anticodons. And again, we use the same base pairing rules. A pairs with U. Remember, we don't have T. C pairs with G. I don't know where my U disappears. And of course, T pairs with A. Okay, so here's our transfer or tRNA with anticodon over here and amino acid attachment site. And here's amino acid is attached. In this example, it's methionine. Translation. To translate the information from mRNA to amino acid sequence or protein, cells use genetic code. The genetic code is a set of instructions that direct the translation of messenger RNA into 20 amino acids, the basic units of protein in living cells. Genetic code is made up of codons, which are 3-letter chain of nucleotides. Each codon codes for one specific amino acid. I know it's a lot of information that you like what it's all about, but let's see how next slides can help us to understand. So first, let's look at this codon. So messenger RNA is a nucleic acid. So all nucleic acids are polymers of nucleotides, right? So here's one nucleotide, here's another nucleotide, another, right? So they're polymers of these nucleotides. RNA is the same as the DNA, has 4 different nucleotides. Let's say this is U, this is A, this is C, this is G, this is U again, and this is A again. Right? The difference is RNA doesn't have any T, but this is our 4 different nucleotides. And so that's a messenger RNA. Now our transfer RNA look like this. So let me remove this. Okay, so here's our transfer RNA. So over here I will have amino acid attached, but on a transfer RNA I have anticodon. And this anticodon corresponds with the codon of messenger RNA. So that's a messenger RNA, and this is tRNA. So let's say if on my messenger RNA I have codon, for example, I have C and U. C and U, right? So that's my codon. That what would be my anticodon? Well, because I have C here, here I would have G, here I would have G, and because I have U over here, I will have A on this side, right? Because it's a base pairing rule. So now only the tRNA that has this anticodon will, will pair with this codon. So they need to pair together. They need to match. If they match, then this is the right amino acid. So I can see what amino acid I code by CCU. So you look here, CCU is proline. So that needs to be amino acid proline. So only this amino acid will match this codon, right? This tRNA with this amino acid proline attached will match this codon. This is how you read this genetic code. So you see at the first letters. So here's the first letter, UCAG. Here's the second. So that's the first letter. This is the second letter, and this is the third letter, right? So for example, AUC. That's amino acid threonine that it codes for, right? This GUU codes for valine, and so on. Okay, so messenger RNA, and you see our nucleotides. So these letters over here, those are nucleotides that make messenger RNA. So we have AU, GG, G, CU, and so on. Now, your cells reading it by codons. So each codon made of 3 letters. So codon number 1 is made of A, U, and G. Codon number 2 is made of G, G, C, right? So here's our codons, right? And tRNA gonna specifically bring the amino acid that is code for this specific codon, right? Because AUG code for methionine. So the first amino acid gonna be methionine. GGC code for glycine. So the second amino acid will be glycine. UCA code for serine. So the third amino acid gonna be serine, and so on until we get to the codon number 7, that is UAA, and it's a stop codon. It does not code for any amino acid. It just means transcription stops. I'm sorry, translation stops. Right, so here then peptide bonds form between amino acid, and we have primary structure of a protein. So translation also has 3 parts: initiation, elongation, and termination, and it's similar to the transcription. Transcription has the same initiation, elongation, and termination. So you can see elongation over here, right there. That's our initiation, and this is a termination. So initiation, we assemble ribosome, messenger RNA, and the first tRNA, and ribosome starts making this protein, synthesizing this protein, and protein getting longer and longer and longer and longer. That's why it's elongation. And finally, when ribosome reaches stop codon, so some here, over here, that's a stop codon on the messenger RNA, it's terminate translation, and we have our newly synthesized protein. So initiation brings together messenger RNA, first amino acid, with it attached tRNA, and 2 subunits of the ribosome. Right, so here's our initiation. We have subunits of ribosome, right? We have messenger RNA, and we also need our first tRNA. And you can see this first tRNA, it needs to be just specifically this one. It cannot be any other tRNA because it must match. Codon need to match with anticodon. So A with U, U with A, right? G with C. So if you have this match, this tRNA gonna park right on this side, and that will be our first amino acid over here. Right, so hopefully you guys saw this animation. The anti-codon of the incoming tRNA pairs with the mRNA codon at the A side of the ribosome. The ribosome catalyzes the bond formation between the two amino acids, and the P side tRNA leaves the ribosome. The tRNA carrying the polypeptide moves from A to the P side. Right, so that's okay, so this is large subunit, small subunit. This is A side. This one is P side, right? And this is our messenger RNA. So here's our instruction coded in this sequence of nucleotide. So the only specific tRNA, so that's our specific tRNA that parks, right, and brings amino acid and ribosomes and synthesize these bonds, covalent bonds between the peptide bonds, covalent bonds, peptide bonds between amino acids, so we can make protein. Right, so here let's look again. So here's our initiation process, right? And then new tRNA is coming. Okay, so here's our tRNA, and it has amino acids attached. But it's only gonna park in this A side if this codon matches with this anticodon. C with G, U with A with U, it matches, then it can park. Right. So here it parks. Now ribosome making these peptide bonds between two amino acids. And now this tRNA gonna leave. And ribosome will shift one codon to the right. So this one, let me do it this way. So what's gonna happen with this tRNA? This tRNA gonna leave, and this tRNA gonna move to the P side. Now pay attention. Now this second tRNA gonna park here. So we have like shift here. This one, number one, gonna leave. tRNA number one gonna leave. tRNA number two gonna move to the P side, and tRNA number three gonna move over here to A side. Right, so this one is leaving. Now this one will move here, and this one will move right there. So now we have 1, 2, 3 amino acids already attached. And I want to remind you that protein is just a sequence of amino acid. Right, so all this long amino acid, 1, 2, 3, and so on, that's what our protein is. So we have so far 3 amino acids already attached. So here's another tRNA is coming, so you can check yourself. So what's gonna happen now? So this tRNA gonna leave. This move to A side, P side, and this move to A side. So elongation continues until a stop codon reaches the ribosome's A side. The completed polypeptide is freed, and the ribosome splits back into its subunits. Okay, so finally we would have that's the chain amino acids over here. This one has 200 amino acids, right? That's our last tRNA, and this is our A side, and it has a stop codon. So there is no any other ribosome that can park here. So our translation is done. So we have termination. The end product of protein synthesis is the primary structure of a protein. Primary structure of a protein is a sequence of amino acids bonded together by peptide bonds. So here's our sequence of amino acid, bounded by these peptide bonds. Now this is a whole process put together in one slide. So we have DNA. So here's our DNA. DNA molecule is this blue one, double helix. That's a process of the transcription, and transcription is making of messenger RNA, then RNA processing, cap, tail, introns cut out, yes, introns, exons put together again, so they connected. Messenger RNA leaves the nucleus. Then we have translation, initiation of the, so here we have some attachment. So we skip this part, but we get straight to step number 4, initiation of translation, elongation of translation, and termination of the translation. So pretty much these parts dissociate, and here's our protein, synthesized protein. Okay, so now what is mutation? Mutation is any change in the nucleotide sequence of DNA. So for example, if our original DNA looked like this, and if during the process of DNA replication, enzyme, the DNA polymerase, put a wrong nucleotide, like over here, we were supposed to have T, but we have C, so that's a new DNA. If we have different nucleotide, we call it mutation. We have different types of mutations, but they can be divided into 2 general categories: nucleotide substitution and nucleotide deletion or insertion. And substitution is a replacement. You substitute one nucleotide with another nucleotide. Deletion and insertion is the loss or addition of nucleotide. And deletion and insertion, they lead to the phenomena that called frame shift. Right? So the reading frame is shifted, and it leads to disastrous effects. So over here you can see the nucleotide substitution. So that's a mutation. So that's a normal DNA for hemoglobin synthesis. And hemoglobin is a protein found inside the red blood cells that carries oxygen all through your body. So that's a normal DNA. And this is the DNA of a person who has mutation. Instead of T, you see right here, this person would have A, adenine, instead of thymine. And of course, we go from DNA to RNA, and from RNA to proteins. And because we have a different nucleotide here on the mRNA, we also have different nucleotide. Now we have U, not A. And because of that, we changing one amino acid. But changing one amino acid in a protein molecule, in a hemoglobin protein molecule, makes actually the whole molecule look different, right? And if this is your normal red blood cells, then a sickle cell, they will look like this. So that's a normal cell and your sickle cell, sickle-shaped red blood cells. So just because of one different protein, the whole protein, just because of the difference in one amino acid, difference in one amino acid in the molecule of hemoglobin looks so different that it changes the shape of red blood cells. And this condition is called sickle cell anemia. So again, this is our point mutation because it's only one. So there is mutation in one specific point, right? Right here. That's point mutation. And it's nucleotide substitution because we substitute T with A. In this example, you can see deletion. So here's a normal gene, and in this example, because I see U, That's already messenger RNA. And now we lost one nucleotide. And one nucleotide is lost. Cells still, they read this information in the nucleotides of 3. So that's the first 3 nucleotides, that is methionine. This is 3 nucleotide, lysine, leucine over here, right? So we're changing all the amino acid after this mutation. So here's where mutation happens. So all these amino acids now are different. And the same gonna happen with the insertion. When we insert one extra nucleotide, right, the whole chain of amino acid here is now different. I wanted to give you example here, just very quick analogy. Or let's go to the next slide and see how it works. Yeah, so here's our summary of the mutations. So summary of the mutation, you have substitution and you have insertion or deletion. And with substitution, mutation can be silent. When we don't change amino acid, it can be missense mutation. When we just swap one amino acid with another amino acid, or it can be nonsense mutation when we get to the stop codon. And insertion and deletion, this mutation can alter the triplet grouping of codons and generally change the amino acid sequence. So the examples that I wanted to give you would be, let's say, if you have this phrase, the cat ate the rat. So this is some type of information, and we read it by the triplet grouping, right? So every word is made of 3 letters, right? And this is how we read it. Now imagine if we have, I'm just going to show you one example. If we have deletion, if this letter E is deleted, then your cells gonna still read it in a triplet grouping. But this is how they gonna group these letters. So they still have T and H, right? But this is only 2 letters, so they're gonna add C over here. Now this is a triplet. Now this C is added to this word. Now what is left? A and T, but this is again only 2 letters, so we need to add one more, right? So this letter is added to this triplet. Now we have TE, and again we need one more, so we're adding this T, and then HER, and then A and T, what is left. So now that's your new information that pretty much has no sense at all. So that's just an analogy to show you that when nucleotide is deleted or it's inserted, so we add extra letter or if we delete one letter, we shift the whole reading frame. And because here information doesn't make sense for you, right? The same information coded in a messenger RNA would not make sense for your cells. And that would be this insertion or deletion mutation, and this can lead to devastating consequences. Okay, so that's it for this chapter, and hope it was helpful.